Welcome back to the deep dive. Today we are taking on, well, a really big one, human reproduction. It's a topic that, affects everyone, but the details are incredibly intricate.
They really are.
And our mission today is pretty clear. We're aiming for a comprehensive deep dive, pulling out every essential detail, structure, function, hormones directly from the NCERT material. The goal is for you to leave with a complete, integrated understanding.
Exactly. And it's so important to approach this material with precision. You can't really understand reproduction without integrating the anatomy, the chemistry, the timing. It all has to fit together.
Right. It's not just a list of parts.
No, definitely not. We need to explain how these systems cooperate, from, say, the structure of the reproductive tracts right through to the really meticulous timing of all the hormonal cycles.
Okay, so let's start right at the beginning.
Yeah.
What are the absolute baseline facts the NCERT establishes? The core biological attributes we need to grasp first.
Well, the NCERT makes it clear from the outset. Humans are fundamentally sexually reproducing organisms. And, very importantly, we're viviparous.
Viviparous, giving birth to live young. That's a key distinction, isn't it? Sets us apart from, say, birds or reptiles laying eggs.
It's a major evolutionary strategy, yes. And another crucial point the NCERT makes is about timing. None of these complex processes, creating gametes, fertilization, pregnancy, birth, none of it happens right away.
It's all delayed until puberty.
Exactly. Everything waits until the individual reaches puberty. Puberty is like the activation switch. Physiological changes, hormonal shifts, they bring the whole reproductive system online from a dormant state into its full working capacity.
Got it. Now, the NCERT breaks down the entire reproductive life cycle into a sequence, almost like a flowchart. You mentioned 7 key events. I think tracing these would give us a really good roadmap for this whole discussion.
That's a good way to look at it. So first up, you have gametogenesis.
The making of the sex cells, sperm and ovum.
Precisely. Then, once you have the gametes, you need insemination. That's the transfer of sperm into the female reproductive tract.
Which obviously sets the stage for the main event.
Which is fertilization, the actual fusion of sperm and ovum. This immediately leads to the formation of a single cell, the zygote.
Okay, single cell formed. What's next?
Well, that zygote doesn't just sit there. It has to start dividing rapidly, developing into what's called the blastocyst.
Little wall of cells.
Essentially, yes. And step 5 is absolutely critical for the pregnancy to continue: implantation. The blastocyst has to successfully embed itself into the wall of the uterus.
If that happens.
Then we enter gestation. That's the whole period of embryonic and fetal development, typically around 9 months in humans.
And the final step.
The sequence culminates in parturition, childbirth, the delivery of the baby.
Gametogenesis, insemination, fertilization, zygote, blastocyst development and implantation, gestation, parturition. That's a fantastic framework. But the NCERT throws in a really fascinating difference right near the start, doesn't it? A major contrast in the lifelong production of gametes between males and females.
It really does. It's perhaps one of the most striking biological differences in the whole system. In males, the process of sperm formation, spermatogenesis, it starts at puberty, as we said.
And then
it just continues pretty much throughout their entire lives. Even into old age, men continue to produce sperm.
But for females, it's a completely different story.
Completely different. The NCERT is very clear here. Ovum formation, or ootogenesis, it actually begins before birth during fetal development, and then, crucially, it completely stops around the age of 50.
That point being menopause.
That's menopause. So you have this finite, non-replenishable supply of potential ova established before birth versus the continuous production in males. It fundamentally shapes the entire female reproductive strategy.
Wow. Okay, that's a massive difference to keep in mind. Let's dive into the architecture then. Part 1, the systems. Starting with the male reproductive system, location, NCERT says, is the pelvis region.
That's right, pelvis region. And it includes the primary organs, the testes, plus what are called accessory ducts, accessory glands, and the external genitalia.
Now, you mentioned the testes. Their location is unusual, isn't it?
It's absolutely key to their function. The testes are located outside the main abdominal cavity. They're held within a pouch of skin called the scrotum.
And this isn't just random anatomy. There's a very specific reason for this external placement.
A crucial reason. It's all about temperature control. Think of the scrotum as a natural thermostat.
Because sperm production is temperature sensitive. The NCERT gives a specific range, doesn't it?
It does. For spermatogenesis, the actual production of viable sperm to happen correctly, the scrotum has to maintain a temperature that's about 2 to 2.5 degrees Celsius lower than the normal internal body temperature.
2 to 2.5 degrees cooler.
Exactly. If the testes were inside the abdomen at core body temperature, sperm production just wouldn't work properly. That slight temperature drop is absolutely essential. It's a non-negotiable requirement.
Okay, so inside this temperature-controlled sac, what's the structure of the tests themselves?
Well, in an adult, each testis is kind of oval-shaped, roughly 4 to 5 centimeters long, maybe 2 to 3 centimeters wide. It's covered by a tough, dense outer layer.
Right.
And inside, the NCERT describes about 250 distinct compartments, like little rooms almost. These are called testicular lobules.
250 in each testis.
Wow. And inside these lobules is where the action happens.
That's the factory floor. Each lobule contains between 1 and 3 highly coiled, tiny tubes. These are the seminiferous tubules. This is precisely where sperms are produced.
And the lining of these tubules is important. Contains specific cells.
2 absolutely vital types of cells line these tubules. First, you have the male germ cells, also called spermatogonia.
These are the precursors to sperm.
Exactly. They are the diploid cells that will undergo meiosis to eventually become sperm. And the second cell type lining the tubules are the Sertoli cells.
What's their role? Think of Sertoli cells as the support crew, or maybe the nurse cells. They provide essential nutrition and various factors needed by the developing germ cells as they mature into spermatozoa. Without Sertoli cells, the germ cells wouldn't survive or develop correctly.
So tubules are the production lines, Sertoli cells are the support staff.
Hmm?
What about the spaces between the tubules?
Yes. The regions outside or between the seminiferous tubules are called the interstitial spaces. These spaces contain small blood vessels, immune cells, and crucially, the Leydig cells.
Leydig cells, also called interstitial cells.
Correct. And these Leydig cells are the hormone factories of the testes. They synthesize and secrete the male sex hormones, the androgens.
Primarily testosterone, I assume?
Primarily testosterone, yes. And testosterone does more than just drive sperm production. It also maintains the accessory ducts and glands, secondary sexual characteristics. It's the main hormonal driver for the whole male system.
And its production is controlled?
Oh, yes. Very tightly controlled. It's regulated by hormones from the pituitary gland, specifically luteinizing hormone, or LH. LH tells the Leydig cells to produce androgens. It's part of a feedback loop involving the brain and pituitary.
Okay, that makes sense. So sperm are produced in the seminiferous tubules. How do they get out? We need to trace the transport route, the accessory ducts.
Right. It's quite a specific pathway. Think of it like a plumbing system designed for transport and maturation. From the seminiferous tubules, the sperm first move into a network of tubules within the testes called the rete testes.
Rete testes, okay.
From the rete testes, they pass through a series of fine ductules called the vasa efferentia. These ducts leave the testis itself and open into the next major structure.
The epididymis.
Exactly, the epididymis. This is a long, highly coiled tube located on the posterior or back surface of the testes. It's a really important site.
Why is that? Storage.
Storage, yes, but also maturation. Sperms spend time in the epididymis undergoing final maturation processes, gaining motility, the ability to swim properly, and the capacity to fertilize an egg.
Okay, so they mature in the epididymis. Where next?
From the tail end of the epididymis, they enter the vas deferens. This is a muscular tube, also called the ductus deferens.
And this is the one that travels up out of the scrotum.
That's the one. It ascends from the scrotum, passes into the abdominal cavity through the inguinal canal, and then loops up and over the urinary bladder.
Right.
Near the bladder, the vas deferens receives a duct from one of the accessory glands, the seminal vesicle, and together they form the ejaculatory duct.
Okay, ejaculatory duct. And that leads.
That short duct passes through the prostate gland and opens into the urethra. The urethra, of course, starts at the urinary bladder and runs through the penis to the outside opening, which is called the urethral meatus.
So the urethra serves both urinary and reproductive functions in males.
Correct. It's a shared pathway for urine and semen, though obviously not at the same time.
Now, you mentioned the seminal vesicles contributing a duct. Let's talk about these accessory glands. Their secretions form the seminal plasma, right?
Yes, the fluid component of semen. The NCERT lists 3 main types of accessory glands. There are the paired seminal vesicles, a single prostate gland, which the urethra passes through, and the small paired bulbourethral glands, also called Cowper's glands.
And what's in their combined secretions? What makes up this seminal plasma?
It's a complex mixture, but the NCERT highlights a few key components. It's rich in fructose.
Sugar for energy.
Exactly. Fructose is the main energy source for sperm motility. It also contains calcium ions and certain enzymes. These secretions provide the transport medium, nourishment, and protection for the sperm during their journey.
Makes sense. They need fuel and a vehicle. Any other specific functions mentioned?
Yes, the NCERT specifically notes that the secretions of the bulbourethral glands play a role in lubrication of the penis, particularly during sexual arousal, preparing for intercourse.
Right. And that brings us to the external genitalia itself.
Yeah.
The penis.
Yes, the penis is the external male reproductive organ. It's made of special erectile tissue that allows it to become enlarged and rigid erection, which facilitates insemination, the delivery of semen into the female reproductive tract.
And the tip.
The enlarged end or tip of the penis is called the glans penis. It's typically covered by a loose fold of skin called the foreskin or prepuce, although this may be removed by circumcision.
Okay. That covers the male system's architecture and plumbing pretty thoroughly. Let's switch gears now to the female reproductive system. The NCERT describes this as a really integrated system, doesn't it? Connecting reproduction with, well, childcare through the mammary glands.
Absolutely. It's a holistic system. The main components are a pair of ovaries, the oviducts, the uterus, cervix, vagina, and the external genitalia. And yes, functionally integrated with the mammary glands for post-birth nourishment.
Let's start with the core, the ovaries, the primary female sex organs.
Right. Analogous to the testes in males, the ovaries have two main jobs: producing the female gamete, the ovum or egg, and secreting the primary female steroid hormones, mainly estrogen and progesterone.
Where are they located?
They're located in the lower abdomen, one on each side. They're relatively small, maybe 2 to 4 centimeters long, almond-shaped. They're held in place by ligaments connecting them to the pelvic wall and the uterus.
And internally, what's the structure?
Each ovary is covered by a thin layer of epithelium, which encloses the main ovarian tissue called the stroma. The stroma is divided into two distinct regions.
Okay.
There's an outer region, the peripheral cortex, which is denser. This is where the action happens in terms of egg development. It contains the ovarian follicles in various stages of growth. And then there's an inner region, the medulla, which is looser connective tissue containing blood vessels and nerves.
Got it. Cortex for follicles, medulla for support. Now the accessory ducts, starting with the tubes that connect, or rather, lie near the ovaries, the oviducts.
Correct. Also known commonly as the fallopian tubes. There are two, one associated with each ovary, though not directly attached. They're about 10 to 12 centimeters long.
And they have distinct parts.
Yes. The part closest to the ovary is a wider, funnel-shaped opening called the infundibulum.
Funnel-shaped.
And the edge of this infundibulum has these delicate, finger-like projections called fimbria.
Fimbriae. They look like they're designed to catch something.
That's exactly their job. Around the time of ovulation, when the ovary releases an egg, the fimbria move and sort of sweep over the surface of the ovary to help collect the released ovum and guide it into the opening of the fallopian tube.
So they prevent the egg from getting lost in the abdominal cavity. Clever design. What's next along the tube?
The infundibulum leads into a wider, longer section of the oviduct called the ampulla.
Ampulla. And this part is significant because
this is the most common site of fertilization.
Yeah.
If sperm are present when the ovum arrives here, this is typically where the fusion happens.
Okay, ampulla for fertilization. Then the tube narrows.
Yes. The final section is a narrow part called the isthmus. The isthmus has a thicker wall, and it's the part that connects directly to the next major organ, the uterus.
The uterus or womb, single structure, right?
Yes, a single, hollow, muscular organ, typically described as being shaped like an inverted pear. It's located in the pelvis, between the bladder and the rectum, and it's supported by ligaments attached to the pelvic wall.
And its lower narrow part opens into the vagina.
Correct. The lower constricted part of the uterus that opens into the vagina is the cervix. The cavity inside the cervix is called the cervical canal.
And the NCERT mentions the birth canal.
Yes. The cervical canal, along with the vagina, forms what's referred to as the birth canal, the passageway through which a baby is delivered during childbirth.
Let's talk about the uterine wall itself. The NCERT describes 3 distinct layers, and they seem really important functionally.
Absolutely crucial. The outermost layer is a thin membranous covering called the perimetrium.
Okay, outer layer.
Then there's the middle layer, the myometrium. This is a very thick layer composed of smooth muscle.
Myo meaning muscle, its function.
This is the powerhouse for childbirth. The myometrium exhibits extremely strong coordinated contractions during labor parturition, which are necessary to expel the baby.
And the innermost layer, the one involved in the monthly cycle.
That's the endometrium. It's the inner glandular lining of the uterine cavity. This layer is incredibly dynamic. It undergoes cyclical changes throughout the menstrual cycle thickening, becoming rich in blood vessels in preparation for potential implantation of a fertilized egg.
And if implantation doesn't occur,
Then much of the endometrium breaks down and is shed, resulting in menstruation. We'll definitely come back to that cycle.
right. Okay, before we get to the cycle, let's quickly cover the female external genitalia, collectively called the vulva.
Okay, the NCERT lists the main structures. You have the mons pubis, which is a cushion of fatty tissue covered by skin and pubic hair lying over the pubic bone.
Then the labia.
Yes, extending down from the mons pubis are the labia majora, which are larger, fleshy folds of tissue that enclose the other structures. Then, paired folds of tissue under the labia majora are the labia minora.
Okay, and inside those.
The opening of the vagina is often partially covered, especially in virgin individuals, by a thin membrane called the hymen.
The hymen. Now, the NCERT includes a really important clarification here, doesn't it? About its significance, or lack thereof.
It does, and it's a crucial point for learners to understand to separate biology from, well, cultural myths. The NCERT explicitly states that the presence or absence of the hymen is not a reliable indicator of virginity or sexual experience.
Why is that?
Because the hymen can be torn or broken through various means other than sexual intercourse. Things like strenuous physical activity, the NCERT mentions sports like horseback riding or cycling or sudden falls, jolts, even sometimes the insertion of a vaginal tampon.
So scientifically, its state proves nothing about sexual history. That's a vital piece of information.
Absolutely vital. And finally, among the external genitalia, there's the clitoris. It's a tiny, finger-like structure located at the upper junction of the two labia minora, above the urethral opening. It's developmentally homologous to the penis and is highly sensitive.
Okay, and the last part of the female system, functionally integrated, is the mammary glands.
Right. Paired structures, the breasts, which contain the mammary glands. These are characteristic of all female mammals, designed for producing milk to nourish offspring.
What's inside them?
Each breast contains glandular tissue and a variable amount of fat. The glandular tissue itself is organized into about 15 to 20 sections called mammary lobes.
Lobes. And within the lobes.
Within each lobe, there are clusters of cells called alveoli. These alveolar cells are the actual milk-secreting units. They produce milk, which is then stored in the central cavities, or lumens, of the alveoli.
So milk is made in the alveoli. How does it get out? There's a duct system here too, presumably.
Yes, another network of tubes. The alveoli open into tiny mammary tubules. The tubules from each lobe join together to form a mammary duct.
Okay.
Several mammary ducts then converge to form a wider structure called a mammary ampulla. This ampulla serves as a small reservoir. And finally, the ampulla connect to lactiferous ducts, which lead directly to the surface of the nipple, through which milk is released during lactation.
Alveoli to tubules to duct to ampulla to lactiferous duct to nipple. Got it. A clear pathway.
A very clear and efficient pathway for milk delivery.
Right, so that's the architecture of both systems laid out. Now we need to get into the actual process of creating the gametes, part 2. Gametogenesis. Let's start with the male side again. Spermatogenesis, occurring in the testes, kicking off at puberty.
Correct. It all begins with the immature male germ cells, the spermatogonia, which are present in the walls of the seminiferous tubules. These are diploid cells, meaning they have the full set of 46 chromosomes.
And they multiply first.
Yes, they undergo mitotic divisions to increase their numbers. Some of these spermatogonia will then grow and differentiate to become primary spermatocytes. These are still diploid, 46 chromosomes.
Okay, primary spermatocytes. Now comes the crucial division to have the chromosome number, right? Meiosis.
Exactly. The primary spermatocyte enters meiosis 1. This is the reductional division. It results in the formation of two equal-sized haploid cells called secondary spermatocytes. Each of these now has only 23 chromosomes.
Haploid, half the number. What happens to these secondary spermatocytes?
They very quickly enter meiosis 2. This division is more like mitosis. It doesn't reduce the chromosome number further, but separates the sister chromatids. This results in 4 equal-sized haploid cells called spermatids, still 23 chromosomes each.
So one diploid primary spermatocyte yields 4 haploid spermatids.
Correct. But these spermatids aren't functional sperm yet. They're basically just round cells. They need a major transformation.
And that transformation has a name.
Yes, it's called spermiogenesis. This is the process where the spermatids differentiate and change shape dramatically. They develop the characteristic sperm structure, the head with the nucleus and acrosome, the midpiece with mitochondria, and the long tail for motility. They transform into spermatozoa, or sperms.
So spermiogenesis is the makeover and the final step, getting released.
Right. After spermiogenesis, the sperm heads are typically still embedded in the Sertoli cells for support. The final release of these mature sperm from the Sertoli cells into the lumen of the seminiferous tubule is called spermiation.
Spermatogenesis, spermiogenesis, spermiation. Got the sequence. Now, this whole complex process must be under tight hormonal control. You mentioned LH before.
Absolutely. The NCERT outlines a clear hormonal cascade. It all starts at puberty with an increase in the secretion of a hormone from the hypothalamus in the brain called gonadotropin-releasing hormone, or GnRH.
GnRH from the hypothalamus. That's the top-level command.
That's the trigger. GnRH then acts on the anterior pituitary gland.
Okay, pituitary gets the message, and it responds how?
It responds by secreting 2 key hormones called gonadotropins. These are luteinizing hormone, LH, and follicle-stimulating hormone, FSH.
LH and FSH. And they target different cells in the testes.
Precisely. LH primarily acts on the Leydig cells, those cells in the interstitial spaces we talked about.
Right, the androgen producers.
Exactly. LH stimulates the Leydig cells to synthesize and secrete androgens, mainly testosterone, and it's the androgens, particularly testosterone, that directly stimulate and maintain the process of spermatogenesis.
Okay, so LH drives testosterone, which drives sperm production. What about FSH?
FSH acts mainly on the Sertoli cells, the nurse cells within the seminiferous tubules.
What does it tell them to do?
FSH stimulates the Sertoli cells to secrete various factors that are believed to help specifically in the process of spermiogenesis, that final maturation and shaping of the spermatids into spermatozoa. So both LH and FSH are essential, working on different targets to ensure the whole process runs smoothly.
A coordinated hormonal effort. Makes sense. Now let's look at the end product itself, the structure of a sperm. Microscopic, but incredibly specialized.
Highly specialized. The NCERT describes it as consisting essentially of a head, a neck, a middle piece, and a tail. And the whole thing is enveloped by a plasma membrane.
The head contains the important cargo.
Yes. The head contains the elongated haploid nucleus carrying the father's genetic contribution, 23 chromosomes. And covering the anterior or front part of the nucleus is a very important cap-like structure called the acrosome.
Acrosome. What's its function? Sounds important.
It's critical for fertilization. The acrosome is packed with enzymes, often called acrosomal enzymes like hyaluronidase and acrosin. These enzymes are released when the sperm reaches the egg and help it penetrate the protective outer layers of the ovum.
So it's like the drill bit allowing entry. What about the middle piece?
The middle piece is essentially the engine room. It contains numerous mitochondria tightly coiled around the axial filament.
Mitochondria for energy.
Exactly. These mitochondria provide the ATP, the energy currency, needed to power the movement of the tail. The tail's lashing movement provides the motility, allowing the sperm to swim towards the egg.
Head for genetics and entry, middle piece for power, tail for movement. A perfectly designed vehicle.
A remarkable microscopic machine.
And the NCERT links this structure to fertility benchmarks, mentioning the sheer numbers involved. What are the stats?
Right. During a single ejaculation, a human male typically releases somewhere between 200 to 300 million sperms. It's an incredible number.
Why so many? Is the journey that perilous?
Extremely perilous. Only a tiny fraction ever reach the vicinity of the egg. And for normal fertility, the NCERT specifies that certain quality standards must be met. At least 60% of the sperm in an ejaculate must have normal shaven size.
60% morphology, okay?
And critically, at least 40% of them must show vigorous motility. They need to be strong swimmers. If these percentages drop significantly, fertility can be impaired. It highlights the intense selection pressure.
Wow. Okay, so that's sperm production and structure. Now let's turn to the female side. Oogenesis, the formation of the ovum and the ovaries. And the key difference here, as you mentioned earlier, is the timing. It starts much earlier.
Dramatically earlier. Oogenesis is initiated during embryonic development itself. While the female fetus is still developing in the womb, Millions of eudagonia, the gamete mother cells, are formed within the fetal ovary.
Millions formed before birth. And the crucial point.
The absolutely crucial point is that no more eudagonia are formed or added after birth. A female is born with her lifetime supply of potential egg cells.
That's fundamentally different from the continuous production in males. So what happens to these eudagonia formed in the fetus?
They start the process of meiosis, but they don't complete it. They enter prophase 1 of meiosis 1, and then they just stop. They get arrested or paused at this stage. At this point, they are called primary oocytes.
Arrested in prophase 1. And what happens around them?
Each of these primary oocytes gets surrounded by a layer of flattened supportive cells called granulosa cells. This entire structure, the primary oocyte plus the surrounding single layer of granulosa cells, is called a primary follicle.
So the fetal ovary is packed with primary follicles containing primary oocytes stuck in meiosis 1.
Exactly. Millions of them initially. However, a huge number of these follicles degenerate during the period from birth through childhood up to puberty. This process is called atresia.
So the numbers drop significantly.
Massively. By the time a female reaches puberty, the NCERT states that only about 60,000 to 80,000 primary follicles are left remaining in each ovary. Still a lot, but far fewer than the millions at the start.
60 to 80,000 potential eggs per ovary at puberty. Okay. What happens to these primary follicles after puberty begins?
With the onset of puberty and the cyclical hormonal changes, these primary follicles start to develop further. The primary follicle grows larger, the granulosa cells multiply, forming more layers around the oocyte, and a new outer layer called the fascicia develops. This stage is now called the secondary follicle.
Primary follicle becomes secondary follicle, more layers. What's next?
The secondary follicle continues to grow and develops further into a tertiary follicle. The key characteristic feature of a tertiary follicle is the appearance of a fluid-filled cavity within the granulosa layers called the antrum.
Antrum, the fluid cavity. That's the identifier for a tertiary follicle.
Yes. Also, the theca layer differentiates into two distinct layers. An inner theca interna, which is vascular and glandular, and an outer theca externa, which is more fibrous.
And something important happens to the oocyte itself at this stage.
Critically important. It's at this tertiary follicle stage that the primary oocyte inside completes its long-delayed meiosis 1.
Finally finishes meiosis 1, and this division is unequal.
Very unequal. The division results in the formation of one very large cell, the haploid secondary oocyte, and one tiny little cell called the first polar body.
Why the size difference?
The secondary oocyte retains almost all of the nutrient-rich cytoplasm from the primary oocyte. This is important because it provides the stored food reserves needed for the early embryo if fertilization occurs. The first polar body is essentially just discarded nuclear material. It may divide again, but eventually degenerates.
Resource conservation for the potential zygote. Clever. So now we have a secondary oocyte, which is haploid, sitting inside a tertiary follicle.
Correct. And the secondary oocyte immediately begins meiosis 2, but it gets arrested again, this time in metaphase 2.
Arrested again, so it doesn't fully complete meiosis yet.
Not yet. It pauses at metaphase 2. Meanwhile, the tertiary follicle continues to mature, growing significantly in size. This final, mature stage is known as the Graafian follicle.
Graafian follicle. That's the one ready for ovulation.
That's the one. Inside the Graafian follicle, the secondary oocyte, arrested in metaphase 2, develops a protective glycoprotein layer around itself called the zona pellucida.
Zona pellucida. We heard about that layer regarding sperm entry.
Yes, it's a key structure involved in fertilization. So the Graafian follicle bulges on the surface of the ovary, and then, typically around mid-cycle, it ruptures. This rupture releases the secondary oocyte, still technically arrested in metaphase 2, from the ovary. This release event is ovulation.
Ovulation, the release of the secondary oocyte from the Graafian follicle.
Oh, okay.
That whole process, from primary follicle to ovulation, is quite involved.
It is. And it happens cyclically after puberty, which leads us nicely into the next major topic.
Right. Part 3, the menstrual cycle and its hormonal rhythm. This is the defining reproductive cycle in female primates, monkeys, apes, and humans.
Correct. The NCERT gives an average interval of about 28 to 29 days, although there's variation. It's the cycle of events from one menstruation to the next.
Now we need the landmarks again. First cycle at puberty is mark.
And the natural cessation of cycles, usually around age 50 or so, is menopause. And as we just discussed, the key event within each cycle is the release of one ovum ovulation, typically around the middle of the cycle.
Okay, the NCERT breaks a cycle down into phases. Let's go through them step by step. Phase 1 starts with the period itself.
Yes, the cycle is conventionally considered to start with the menstrual phase, or menstruation. This phase lasts for about 3 to 5 days on average.
And what's actually happening physiologically?
It's marked by the menstrual flow, which consists of blood, tissue fluid, mucus, and the degenerated cells from the endometrial lining of the uterus. Remember that inner layer we talked
The
about?
one that builds up each month.
Exactly. During the menstrual phase, that built-up lining breaks down because the necessary hormonal support has been withdrawn. The blood vessels rupture, leading to bleeding.
And the underlying reason this happens, the NCERT specifies a condition.
Yes, menstruation only occurs if the ovum released during the previous cycle has not been fertilized. If fertilization and implantation occur, the hormonal signals change, the endometrium is maintained, and menstruation does not happen.
Which is why a missed period is often the first sign of potential pregnancy, though the NCERT notes other causes too.
Right. A lack of menstruation isn't always pregnancy. It can also be due to other factors like stress, poor health, underlying medical conditions, significant changes in routine, etc.
Okay, so after the shedding during the menstrual phase, the body starts rebuilding. What's the next phase called?
This is the follicular phase, also sometimes called the proliferative phase because the endometrium starts proliferating or growing again.
Follicular phase makes sense because the follicles in the ovary are developing too.
Precisely. This phase overlaps with and follows menstruation. During this time, under the influence of pituitary hormones, several primary follicles in the ovary begin to grow, developing into secondary and then tertiary follicles. Usually, only one of these will become the dominant follicle, maturing into the Graafian follicle.
And in the uterus.
Simultaneously, the endometrium, which was shed during menstruation, begins to regenerate and thicken. It proliferates, restoring the lining.
What drives these changes? Hormones, obviously.
Yes, hormones are key. This phase is driven primarily by the gonadotropins from the pituitary FSH and LH. Their levels gradually increase during the follicular phase.
FSH stimulating follicle growth.
Yes, and as the follicles grow, particularly the dominant one, they start producing increasing amounts of estrogen.
Estrogen. So rising estrogen levels characterize the follicular phase.
Yes, and this rising estrogen has multiple effects. It stimulates the proliferation of the endometrium, and it also feeds back to the pituitary, initially inhibiting but later stimulating LH release.
Okay, so follicles grow, endometrium rebuilds, estrogen rises. This leads up to the midpoint event.
Exactly. Around day 14 of a typical 28-day cycle, we hit the ovulatory phase. This is triggered by a dramatic hormonal event,
The LH surge.
the famous LH surge. Both LH and FSH levels peak around mid-cycle, but the LH peak is particularly sharp and pronounced. This rapid secretion and maximum level of LH is the LH surge.
And this surge is the direct trigger for.
It's the direct trigger that causes the mature Graafian follicle to rupture and release the secondary oocyte. In other words, it induces ovulation. The FSH surge might also contribute.
Ovulation happens roughly 24-36 hours after the LH surge starts.
Right.
So the egg is released. What happens to the remnants of the Graafian follicle left behind in the ovary?
They transform. This marks the beginning of the final phase, the luteal phase, also called the secretory phase because the endometrium becomes secretory.
Luteal phase referring to the corpus luteum.
Precisely. The remaining parts of the ruptured Graafian follicle, the granulosa and the ceca cells, undergo a rapid transformation into a yellowish endocrine structure called the corpus luteum, which means yellow body.
And its job. It becomes a temporary hormone factory.
Exactly. The corpus luteum is a temporary endocrine gland, and its main function is to secrete large amounts of progesterone. It also secretes some estrogen.
Progesterone. Why is that so important in this phase?
Progesterone is absolutely essential for maintaining the endometrium. It makes the endometrium even thicker, more vascularized, and more glandular. It becomes secretory, producing nutrients. This prepares the uterus to receive a potential fertilized egg for implantation.
So progesterone basically makes the uterus welcoming and supportive for pregnancy.
That's a good way to put it. It's the hormone of pregnancy maintenance, especially in the early stages.
Okay, so the corpus luteum pumps out progesterone, maintaining the endometrium. What happens if fertilization doesn't occur after ovulation?
If there's no fertilization and implantation, the corpus luteum has a limited lifespan. After about 10-14 days, in the absence of hormonal signals from an implanted embryo, specifically HCG, the corpus luteum starts to degenerate and shrink.
It breaks down.
It degenerates into a scar tissue called the corpus albicans. As the corpus luteum degenerates, the levels of progesterone and estrogen drop sharply.
And the falling hormone levels?
The withdrawal of progesterone support causes the endometrium to break down and disintegrate. The blood vessels constrict, then rupture, leading to shedding, and that's menstruation, marking the start of a new cycle.
The whole cycle resets. Wow. It's a beautifully orchestrated rise and fall of hormones controlling events in both the ovary and the uterus.
It truly is a complex interplay. And before we move on from the cycle, the NCERT also makes a point about practical health aspects.
Ah, yes, menstrual hygiene.
Yes, it stresses the importance of maintaining hygiene during menstruation. This includes regular bathing and cleaning and using clean sanitary protection like napkins or tampons, changing them regularly, perhaps every 4 to 5 hours depending on flow to prevent infections, and also proper disposal of used sanitary products.
Good practical advice integrated there. Okay, let's move to part 4. What happens if fertilization occurs? The journey from fertilization to fetus.
Right. So following sexual intercourse, semen is deposited in the vagina, that's insemination. The motile sperms then have a long journey ahead.
They have to swim.
They swim rapidly, passing through the cervix, up through the uterus, and finally into the fallopian tubes, or oviducts. Their target is the ampullary region of the fallopian tube.
Where the ovum is hopefully waiting or arriving soon after ovulation.
Exactly. And the NCERT reiterates a crucial point about timing here.
That's simultaneous arrival.
Yes. Fertilization can only occur if the ovum and the sperms are transported simultaneously to the ampullary region. This narrow window of opportunity, the lifespan of the egg, about 12-24 hours, and the viable lifespan of sperm in the female tract, up to a few days, explains why not all acts of coitus lead to pregnancy.
Timing is everything. So, assuming a sperm reaches the ovum in the ampulla, what happens at the moment of contact?
The sperm first has to penetrate the outer layer surrounding the secondary oocyte, the corona radiata, layers of follicular cells, and then the zona pellucida. When a sperm makes contact with the zona pellucida layer,
The glycoprotein coat.
yes, that contact triggers the acrosome reaction in the sperm, releasing those enzymes we talked about. These enzymes help the sperm digest a path through the zona pellucida.
Okay, it breaches the zona pellucida. What next?
As soon as the plasma membrane of the sperm fuses with the plasma membrane of the secondary oocyte, it induces critical changes in the oocyte membrane.
Changes to prevent other sperm from entering.
Precisely. It triggers the cortical reaction in the oocyte, releasing substances that make the zona pellucida impenetrable to any other sperm. This is the mechanism that blocks polyspermy, ensuring only one sperm fertilizes the ovum. It's vital.
One sperm gets in, the door slams shut. Got it. What does the sperm's entry trigger inside the oocyte?
The entry of the sperm, specifically its nucleus, into the cytoplasm of the secondary oocyte acts as the trigger for the oocyte to finally wake up from its second arrest. It completes meiosis too.
Ah, it finally finishes dividing.
Yes. The second meiotic division is also unequal. It results in the formation of the mature haploid ovum, now technically called an ootid, though often just referred to as ovum, and another tiny second polar body, which also degenerates.
So now we have the haploid nucleus of the mature ovum and the haploid nucleus of the sperm inside the same cell.
Correct. These two haploid nuclei, called pronuclei, then fuse together. This fusion event, called syngamy, restores the diploid chromosome number 46 and forms the single-celled zygote. Fertilization is complete.
Zygote, the beginning of a new individual. Now, the NCERT uses this point to clarify something about sex determination, addressing a common misconception.
Yes, it's a really important clarification based on simple genetics. Remember, the chromosomal basis of sex in humans is XX for females and XY for males.
Right.
During oogenesis, all mature ova produced by a female will contain one X chromosome, since females are XX.
Okay, all eggs carry an X. What about sperm?
During spermatogenesis, because males are XY, meiosis results in approximately half the sperm carrying an X chromosome and the other half carrying a Y chromosome.
50-50 chance for X or Y sperm.
Exactly. Therefore, the sex of the resulting zygote depends entirely on which type of sperm fertilizes the X-carrying ovum.
So if an X-carrying sperm fertilizes the ovum,
X from sperm plus X from ovum gives XX, the zygote develops into a female baby.
and if a Y-carrying sperm fertilizes the ovum.
Y from sperm plus X from ovum gives XY, the zygote develops into a male baby.
So the conclusion is clear.
Crystal clear, scientifically speaking. The sex of the baby is determined by the chromosome carried by the sperm, which comes from the father, not the mother. The NCERT emphasizes this to counter societal blame often directed at women for the sex of their child.
A crucial scientific and social point. Okay, so the zygote is formed in the fallopian tube. What happens next? Does it stay there?
No, it immediately starts its journey towards the uterus. And as it travels down the fallopian tube through the isthmus, it begins to undergo rapid mitotic cell divisions. This process of early division is called cleavage.
Cleavage. So the single cell divides into
2 cells, then 4, then 8, then 16 cells. These daughter cells produced during cleavage are called blastomeres.
2, 4, 8, 16 blastomeres. Still moving towards the uterus.
Yes. The embryo at the 8 to 16 cell stage, looking like a solid ball of cells, is called the morula, from the Latin for mulberry, which it resembles.
Oh, okay.
The morula continues to divide, and as it reaches the uterus maybe 3-4 days after fertilization, it starts to transform into the next stage, the blastocyst.
Blastocyst. How is it different from the morula?
It develops a fluid-filled cavity inside called the blastocole. And crucially, the blastomeres arrange themselves into 2 distinct cell populations.
2 layers.
Yes. There's an outer layer of cells called the trotoblast, and there's a group of cells clustered together at one pole inside called the inner cell mass.
Trophoblast on the outside, inner cell mass on the inside. What are their fates?
The trophoblast layer is really important for implantation and will later contribute to forming the placenta. The inner cell mass is what will actually develop into the embryo proper, all the tissues and organs of the baby.
Okay, so the blastocyst arrives in the uterus. Now comes that critical step you mentioned earlier, implantation.
Yes. Around day 6 or 7 after fertilization, the blastocyst needs to attach to and embed itself into the wall of the uterus, the endometrium, which has been prepared by progesterone. The trophoblast layer plays the key role here. It adheres to the endometrial lining.
It sticks to the wall.
Yes. And then the uterine cells of the endometrium divide rapidly and grow over, engulfing the blastocyst. So the blastocyst becomes fully embedded within the endometrium. This process is implantation.
And successful implantation marks the beginning of.
It leads to pregnancy.
Right. So implantation is successful. What develops next to support the growing embryo? The placenta.
Exactly. After implantation, finger-like projections start appearing on the surface of the trophoblast. These are called chorionic villi.
Chorionic villi.
These villi grow and extend into the surrounding uterine tissue, the endometrium. They become intricately interlocked or interdigitated with the uterine tissue and the maternal blood vessels. This complex structure formed by the merging of fetal, chorionic villi, and maternal uterine tissues is the placenta.
So it's a combined fetal-maternal organ. What's its main job?
It forms the vital connection between the developing embryo fetus and the mother. Its primary function is to facilitate the exchange of substances. It allows the supply of oxygen and essential nutrients from the mother's blood to the embryo.
And removal of waste.
And enables the removal of carbon dioxide and metabolic waste products produced by the embryo back into the mother's blood for disposal. It acts like the fetal lungs, gut, and kidneys all rolled into one.
How is it connected to the fetus?
The placenta is connected to the embryo fetus by the umbilical cord. This cord contains blood vessels that transport blood between the fetus and the placenta.
Okay, so it's a life support system. But the NCERT also highlights its role as an endocrine organ. It makes hormones.
Yes, this is crucial. The placenta acts as a temporary but very active endocrine tissue, producing several important hormones required to maintain the pregnancy and support fetal growth.
Which hormones specifically?
The NCERT lists several, notably human chorionic gonadotropin, HCG.
HCG, that's the one detected in pregnancy tests.
That's the one. Its main early role is to signal the corpus luteum in the ovary to keep producing progesterone, maintaining the endometrium until the placenta can take over progesterone production itself.
Okay, HEGG. What else?
Human placental lactogen, HPL, which has effects on maternal metabolism and breast development. Also estrogens and progestogens, mainly progesterone. The placenta becomes the main source of progesterone after the first few weeks.
And the NCERT mentions some hormones produced only during pregnancy.
Yes, HCG, HPL, and another hormone called relaxin, which is actually secreted mainly by the ovary, stimulated by HCG later in pregnancy to help relax ligaments, are produced in women only during pregnancy.
Interesting. And other maternal hormones increase too.
Yes. During pregnancy, the levels of several other maternal hormones also increase significantly. Things like cortisol, prolactin for milk production, thyroxine, thyroid hormone. The NCERT states these increases are essential for supporting fetal growth, managing the metabolic changes happening in the mother, and generally maintaining the pregnancy.
A massive hormonal shift overall. Now, while the placenta is forming and working, what's happening to that inner cell mass, the part that becomes the baby?
The inner cell mass starts to differentiate very quickly after implantation. It organizes into the three primary germ
The
layers.
fundamental layers.
Yes, the outer layer is the ectoderm, the inner layer is the endoderm, and shortly after, a middle layer, the mesoderm, develops between them.
Ectoderm, mesoderm, endoderm. And these three layers give rise to everything.
Absolutely everything. Each germ layer is destined to form specific tissues and organs in the adult body. For example, ectoderm forms skin and the nervous system, mesoderm forms muscle, bone, and connective tissues, and endoderm forms the lining of the digestive and respiratory tracts and associated glands. It's the fundamental blueprint.
And the NCERT mentions stem cells here.
Yes, it points out that the inner cell mass contains certain cells called stem cells. These cells are pluripotent, meaning they have the remarkable potential to divide and differentiate into all the various cell types, tissues, and organs that make up the body, a key area of biological research.
Fascinating potential. Okay, let's track the major milestones of development during gestation. The NCERT provides a timeline. What's the first big event?
One of the very first major organs to form is the heart. The NCERT states that after just 1 month of pregnancy, the embryo's heart is formed and starts beating. This heartbeat is often the first sign of a viable pregnancy that can be detected, for instance, by listening with a stethoscope or ultrasound.
Heartbeat at 1 month. Wow. What about by the end of the second month?
By the end of the second month of pregnancy, the developing individual is now generally referred to as a fetus. And importantly, it starts developing recognizable limbs and digits, fingers and toes are forming.
Limbs and digits by 2 months. What marks the end of the first trimester? Around 12 weeks.
By the end of 12 weeks, or the first trimester, most of the major organ systems are formed in a rudimentary way. For example, the limbs and external genital organs are well developed, and it might even be possible to determine the sex externally.
Major systems laid down by 12 weeks. What happens during the second trimester? Any key milestones?
Yes. The NCERT highlights that during the 5th month, the first movements of the fetus can usually be felt by the mother. Also, fine hair starts to appear on the head.
Fetal movement and hair by 5 months. What about the end of the second trimester, around 24 weeks?
By the end of 24 weeks, the body is covered with fine hair called lanugo. The eyelids separate and eyelashes are formed. The fetus is becoming much more developed, although still needs significant maturation.
Okay. And the third trimester?
The 3rd trimester, from about week 25 onwards, is primarily a period of rapid growth and maturation of the existing organ systems, accumulation of fat, and preparation for life outside the womb. By the end of 9 months of gestation, the fetus is generally considered fully developed and ready for delivery.
9 months of incredible transformation, which brings us to the final act. Part 6, parturition and lactation. Let's start with parturition, childbirth.
Right. The average duration of human pregnancy, or the gestation period, is about 9 months. Parturition is the process of expelling or delivering the fully developed fetus from the uterus at the end of this period.
And it involves powerful uterine contractions.
Yes, vigorous contractions of the uterine myometrium are the driving force.
What triggers it all? The NCERT calls it a complex mechanism.
It's described as a complex neuroendocrine mechanism, meaning it involves both nerve signals and hormones. The process is thought to be initiated by signals originating from 2 sources. From the fully developed fetus itself and also from the placenta, these signals together induce mild uterine contractions to begin with. This initial phase is often called the fetal ejection reflex.
Fetal ejection reflex. So the baby and placenta sort of signal, we're ready.
That's the idea. These mild contractions then trigger a crucial hormonal response in the mother.
Oxytocin.
Yes. The nerve signals generated by the uterine stretching and contractions travel to the mother's brain, specifically the pituitary gland, causing the release of the hormone oxytocin.
And oxytocin's effect.
Oxytocin acts directly on the smooth muscle of the uterine wall, the myometrium, causing it to contract much more forcefully.
Ah, so it ramps up the contractions.
Exactly. And here's the clever part. It's a positive feedback loop. The stronger uterine contractions caused by oxytocin stimulate further release of oxytocin from the pituitary.
More contraction leads to more oxytocin, which leads to even more contraction.
Precisely. The stimulatory reflex continues, escalating the strength and frequency of contractions until they are powerful enough to expel the baby from the uterus through the cervix and vagina, the birth canal. This expulsion is childbirth.
And after the baby is delivered?
Shortly after the delivery of the infant, the placenta is also expelled from the uterus. This is sometimes called the afterbirth.
A remarkable feedback loop. driving the whole process. Okay, finally, after parturition comes lactation, milk production.
Right. The mammary glands, as we discussed, undergo differentiation and development during pregnancy, stimulated by hormones like estrogen, progesterone, and placental lactogen. Towards the end of pregnancy, they start producing milk. This process is called lactation.
But the milk produced right at the beginning is special, colostrum.
Yes, the milk produced during the initial few days after delivery is called colostrum. It's often yellowish and thicker than mature milk, and the NCERT stresses that it is absolutely essential for the newborn.
Why is colostrum so vital? What's in it?
Its key benefit is that it's incredibly rich in antibodies, particularly immunoglobulin A, IgA. These antibodies are passed from the mother to the baby and provide crucial passive immunity, helping to protect the newborn from infections during the first vulnerable weeks and months of life. It helps develop resistance.
Nature's first vaccine, in a way.
That's a great analogy. It's packed with protective factors. That's why the NCERT strongly recommends breastfeeding, especially during the initial period of infant growth, to give the baby the benefits of colostrum and continued nourishment and immunity from breast milk.
Excellent. That covers the entire journey as laid out by the NCERT.
From gamete formation right through to postnatal care.
It really has been a comprehensive deep dive. We've traced everything from, the very specific temperature needed for sperm production, right through that incredibly complex hormonal dance of the menstrual cycle, and ending with the neuroendocrine signals triggering childbirth, all following the NCERT's integrated view.
And I think if you take one thing away, it's that integration. You see how structure, like the layers of the endometrium or the seminiferous tubules, is completely linked to function, which is driven by that precise, often cyclical hormonal control like the LH surge triggering ovulation or progesterone maintaining the uterine lining. It all works together.
Absolutely. And then thinking about that aha moment for the learner, that fact from the NCERT that maybe challenges assumptions, what stands out? Maybe the clarity on sex determination or that difference in gamete lifespan.
Both are excellent examples. The fixed versus continuous gamete production is profound, but perhaps the sheer complexity and almost fragility of the hormonal coordination needed for pregnancy and birth is also worth pondering. That positive feedback loop of oxytocin during parturition, for instance.
What about it?
Well, think about it. It's a stimulatory loop that has to build perfectly, reach a climax to achieve expulsion, and then shut down correctly afterwards. The precision required in that final cascade of neuroendocrine signaling to ensure a successful, safe human birth. It really highlights the incredible, intricate control systems operating within our bodies. It's quite something to muse on.
It really is. The complexity is staggering when you break it down.
Indeed.
Well, that seems like a perfect point to wrap up this deep dive.
A good place to pause, definitely.
Thank you for joining with us today. See you next time.
See you next time.